Hi, my name is Pawel Michalak. I run the lab of integrative genome analysis in the division of medical informatics and systems here at Virginia Bioinformatics Institute. My lab focuses on comparative genomics, epigenetics, and evolution. So we are interested in how genomes evolve through time, how they adapt to various stresses. We are not really attached to any particular system, but our two favorite systems are fruit flies, Drosophila, and Xenopus African cloud frogs. Xenopus cloud frogs have a very exciting model system for many reasons. One of them is that they have a unique range of genome sizes. We call it ploidy level. So there are species that are diploid, so they have a genome size like we do, but there are tetraploid species that have duplicated genomes. There are octaploid species and there are dodecaploid species that have more than 100 chromosomes. So it's very interesting to have an animal system like that. It's very common among plants, but it's very rare among animals. So if someone is interested in what happens to genes after a whole genome is duplicated, it's an ideal system. And you can imagine that if there is a gene A after duplication, there are two copies of it. So they're free to diverge. They're free to specialize on different functions. Because of this range of ploidy levels, we have different genome sizes and different copies of the same gene. We can see what happens with all the copies in different species. So another very interesting feature of frogs is the interspecies hybridization. So there are two species, let's say Xenopus laevis and Xenopus mulleri. When they hybridize, they produce F1 hybrids. And what happens there at this nucleus level is very interesting. It's like a dramatic epigenetic phenomenon, very poorly described so far, called nucleolar dominance. Turns out that parental frogs have two nucleoli. This is like a center of ribosomal RNA production. And F1 hybrids have only one. It looks like it's all maternally determined. So ribosomal RNA is only from the mother are expressed. Those copies from the father are suppressed. It's really a dramatic effect because ribosomal RNA is the most abundant RNA in the cell. So there's a mechanism that shuts down multiple copies of ribosomal RNA genes, and only one nucleolus is formed. It's really a very distinct feature. Again, it's poorly understood. It's comparable to like X chromosome silencing in mammalian females.